And our next speaker is Mr. Rajavarman Kittu, and he'll be highlighting and telling us about NGS technology infrastructure in India and its application. To talk about Mr. Rajavarman, he has completed his master's in bioinformatics from Shanmugha Arts, Science, Technology and Research Academy. His expertise lies in bioinformatics, computational biology, genomics, NGS, comparative genomics, cancer genomics, and he has also worked as a senior technical officer, clinical bioinformatician at Kokilaben, Dhirubhai, Ambani Hospital. And we are pleased to have you with us, sir. Over to you.
I'd like to thank Biotech for the given chance. Can you just confirm if you can hear me clearly, plus you can see my screen?
Yes, sir. We can hear you clearly, and also we can see your screen.
Yeah, sure. Pardon me for the noise. I mean, somewhere in the transit. Yes, so far I think we have covered. I just listened the previous talk. I think we went very deep in the COVID sick. Now we are going to switch the gear. So currently we are going to see what is the infrastructure generally needed for the NGS. So I'm just comparing NGS to C. C in a sense the SEAC. So since there is so much of varieties, there's so much of things to explore. We are going to surf in the sea now, right? Literally the ocean. Let's see how it goes forward. So the main thing which you need to know is NGS size. When I say size, it means the file which come out of any given machine is basically different, very different. The file format as well as the individual file sizes, starting from MB to GB. So in that case, the size really matters when it comes to NGS, right? Second one is which machine you are going to handle and what are the applications you do. And the third one is the checklist for readiness. When we say you're going to do the NGS, what are things you need to have in place? So let's go into detail. As I told before, the smaller organism generally gives in MBs, and this, let's say human data, you can go still 3.3 for 1x. So in that case, when we consider for 30x or 1000x, it's going to be bigger and bigger. So this is not just to scare you, rather than to make sure this is really the big data, which is a buzzword, which is happening in the IT industry. It's literally happening in the biology as well, in the genomics field. So it's up to the depth. It's up to us to decide how much depth we are going and how big the data is. Again, the second thing which we need to know very clearly is the data flow. When I say data flow, it means how frequently we are going to run the machine. In essence, what are the samples we are going to load, how many samples we are going to load, how frequently we are going to operate the machine, right? Second one is deep sequencing. As I told, how much depth we are going. Is it like 50x, 100x, or 250x, 500x, or it's more than that? Even sometimes people go for even 10,000x. Based on the question which we are asking, the scientific question we are asking, the depth can go like probably vary, right? And the last one, which is very important, is what kind of panel you're going to run. Is it just a basic panel with a limited number of genes, probably the targeted genes, or the full exome, or the whole genome, or whole exome kind of data, right? So these three things has to be anyway first should be understood. And the next important thing is what are the things we are going to archive. So when I say archive, basically, for any given English data set, we'll be having a raw data, process data, and we'll be going for archive. Apart from that, in the clinical setup, we might even need to do the reanalysis or we need to review the data set. So what do you mean by reanalysis? Suppose down the line, so there are steps where we'll be going for annotation for the given variant. So that will be changing with respect to time for certain number of genes, right? So those kind of cases where reanalysis is important. Based on that, even the therapy recommendation might vary, not in the bigger level, but in the slight deviations can be possible. So in that case, reanalysis is one of the key factors. So we need to be knowing what are the things will be permanently archived and what are the files or probably what are the details we need to have always like in ready, so that we need to go for reanalysis. Apart from when I say review, it means say 5 years you have did some analysis. You want to recollect in the 5 years what are the variants like very commonly occurred from the given data set. And probably you can even find the trendings in the sense recent 2 years. What are the new variants or what are the recent findings are from the given data set. So those kind of repurpose related analysis, this module, like the second part has to be considered. So in that case, when we really go for the data management, we need to know what are the regular exercise and what are the repurposes exercise, right? And which mission. So as we know in the market, there are, when I say market, in this market, there are very huge varieties of machines are available. Based on the needs and the application, we need to be sure about which one we are choosing and whether it is solving all of the questions or not. It is useful to solve all the questions which we ask, right? The first is purpose, as I told. Whether are you asking a basic question or intermediate or advanced? So whether the given technology will help us to answer this one. The second one is sample load. How, as I told before, how frequently you're operating the machine, how much load are you having, like the data load which you're going to have it or handle it. Last one is the capacity, whether are we having high throughput machine or low throughput machine, right? So these are the key points which has to be checked beforehand. I was keep on speaking about the missions. So when I say missions, it means as we can see the varieties starting from 1.2 GB to 6000 GB. So this is a capacity for the single run when we fully utilize the machine to a full extent, right? So as I told before, these are all the let's say low throughput, and this is a mid throughput, and this is a high end, right? And these, where do we use a low throughput machines into? For sequencing the smaller organisms, be it bacterial, virus, fungus, right? Any given smaller organism, except the human, for the whole genome sequencing, it can be used. So particularly for smaller organisms, this sequencing machine is being used, right? And apart from this, if the sample number is less, and we need to have, let's say, as I told before, 0.2 GB, 1.2 GB, right? So those kind of smaller amount of data, if you want to let's say generate, these kind of facilities fair enough, right? When we are going for bigger projects, say population studies or cohort related analysis, those kind of bigger studies, if you want to do, probably we need to have this kind of hierarchy. Even including the COVID sequencing data set, we need to have these kind of missions in place, right? For example, when it comes to human, whole genome sequencing and whole exome sequencing, right? And apart from that, when the sample load is higher, even though it's a smaller organism, but your sample load is very higher. So in that case, probably you'll be using this kind of sequences, right? So this is prone to give high throughput. Let's see what are the applications generally we get out of these machines. Of course, these machines can be used in all kinds of set of clinical, academic, and the research. Currently, what we are seeing is in the clinic and research, it's been widely used. Academy, it's slowly coming up. So we'd like to see more the missions which we discussed in the probably the forthcoming futures, right? Application wise, if you see, as I told before, you name it any given application of genomics, it can be performed with the missions which we discussed. Whole genome sequencing, whole exome sequencing, methylation, single cell, HLA, PGD, PGS, comprehensive panel, targeted panels, RNA. So you name it, any kind of application, everything can be done, right? So, uh, okay, we know about the background now. Can we list out what are the key points to do that we should be having to proceed? So the first one is security. Why? Because there are very sensitive data is being used in the NGS, probably the medical data sets, where we don't reveal much details everywhere. Rather, every all the patient data will be converted in the particular ID, and that will be processed all over. In that case, even the data access will not be given to everybody, since there is a HIPAA accredited certifications are given to the informatics tools. Security is the first concern, right? Second one is user friendliness. Since it's not like everyone is master in the informatics to do the analysis and give the reporting. So in that case, the friendliness is user friendliness is very important. Third one is basically audit trials of file access. Why? When it comes clinical setup specifically, which file is being accessed by whom at what time, that will be traced out, because that's very important thing to back not only the backtrace, even to there are multi-levels of screening will be done to release the report. Even those kind of places, this audit trail is very important, right? And the fourth point is basically manpower. As I told, establishing the full facility, starting from scratch to a reporting solution, is it basically needs a really good number of manpower who is very much skilled. And apart from that, we need to have a good battery backup for the setup, like say to have an infrastructure. And the room temperature should be monitored for 365 days in a year. And dedicated network is needed. Apart from that, good internet speed is needed. Why these are the things are needed? Since we are speaking about 6,000 GB data set from one particular run, you can imagine how long it takes to transfer the data set, process the analysis, and then archiving it. So in that case, these are the basic needs should be in place, right? So say I'm very new, I'm from academic. Since I can't go for all kind of thing. I have only simple laptop. Can I do the NGS analysis? Yes, you can do. Probably if you see there are softwares like Galaxy, and there are even some pipelines which is ready-made available which can be used using even Windows. But typically, if you want to do in-house without sharing data with any of the third party, probably outsiders, in that case, this is a computation which is expected. Like this is a RAM, this 96 processor, probably storage is 50 TB and Linux storage. It's very difficult to get as a workstation. Probably as a server linked with the workstation is good to go with. So in that case, to start with complicated, to answer the complicated question, this kind of computation is expected, right? So I got the facility now. I got the setup now in place. What are all the basic things I need to check to make sure my data is good enough to do the analysis? First thing you need to know is coverage. Like whatever the panels I sequence, have got all the data set in place. Second one is depth. Like whatever the depth I estimated, have I reached the depth? In the sense, how many x depth have you reached? Right? Reads. There are cases where in RNA kind of data set, you have to know how many reads have been generated, whether those are all enough to proceed further. And the third one is machine, as I told, which machine should I process to get these kind of data sets? And sample number, as I told before, how many samples can occupy in one particular batch? If I put this many samples in this particular batch, how much of coverage and depth will I get? So those kind of pre-calculation estimation should be well defined so that the expected results are in place. And files to preserve, as I told before, which are the things should be archived permanently, which are the things should be in the front desk so that anytime you can access. That part should be very much well defined in SOPs. And finally, the process. So before even process, I would say there has to be a testing phase, or probably the beta versions should be there in place to make sure, yes, every individual process is going well without any disturbance, right? Uh, when it comes to research setup, probably open source tool if you plan to use, how does it goes? This is how the overall workflow goes. The literature survey will be conducted based on whatever the tools which is widely being used, and the same tools will be downloaded, and then we'll be standardizing based on the data set and the question which we ask. So when I say standardize, it means fine tuning the parameters. When I say validate, it means same kind of study is being observed from the PubMed publications, and then replicating the same work so that to make sure, yes, my tool is meeting the standards which has been already published. So that is a validation. And finally, the pipeline. Again, every individual application which we discuss have its own pipeline and the steps. Every steps will be validated with the relevant parameter, and then we'll be going for the final product, right? So this is a protocol which is being followed for in-house development. So in that case, the complication is really very high until otherwise you are not skilled. So in that case, there are alternative solutions which is there in market, which is basically the softwares are already feeded in, the parameters are already tweaked, and it's very easy to use. In that case, cloud solutions are available which can be used. Apart from that, there are some servers which comes with FPGA card where all the programmings are already feeded, validated, and standardized. The moment you probably it's been installed from the day one, it can be used. So I'm not going into these two topics much. Why? Because we are mostly going to speak about infrastructure. With that, I'm just closing this talk. If you have any questions, I'd like to take it forward.
Thank you so much, Mr. Rajavarman, for your presentation. If you have any questions for sir, please put them in the chat box and we'll be able to get your questions answered. And also those participants who are asking about the recordings, yes, we'll be sharing with you the recordings very soon. For that, you need to fill out the registration form so that we have your email IDs at least where we can share the recordings with you all. So we'll just put the registration form the list in the chat box, and those who have not registered themselves, they can fill the form. Okay, so sir, I wanted to ask the question, what are the career opportunities with Premas Life Sciences?
Okay, currently, since you took the name Premas Life Sciences, I'd say there are who are PhD and postdoc passed out, they have opportunity for product managers, as I can see, right? Again, not in the informatics perspective, overall, probably the coming up with the new assay, validating studies. So there are multiple openings out there where probably we even, since we are covering all kind of segments in the sense, we train the people in the analysis, we train the wet lab people for preparing the libraries and loading the samples. Plus, we have a specific marketing team who takes care of probably all the marketing activities and the sales team. So in that case, we have opening almost in all the departments at this given point. So if you're asking specifically informatics, I don't. There might be a junior level intern opportunity might be available, but not a full time job. But with other segments, there are full time jobs are there at this time.
Okay, sir. Thank you so much. And the next question is, what could be the possible online resources for NGS-based identification of microbial variety?
Okay, so when you say the database is already in place for almost all the model organisms, the sequencing data, which the process data as well as the sequencing data is in place. Particularly when you say smaller organism, I can think about two things quickly, which is MTB and COVID. COVID data is anyway it's out with GSIDE actually. At the same time, even NCBI SRA has, to name it any kind of application, everything is already in place. So this is two resources which I can think about quickly.
Right, sir. And is Linux a must for NGS bioinformatics? What are your thoughts on it?
Again,
is Linux, yes, sir. The question is Linux a must for NGS bioinformatics?
Okay, is Linux is important? Okay. So I would say Linux is even, I think the voice is breaking. No issues. So the Linux is important. I don't think so it is important because since as I told the last slide, the cloud computing solutions which are coming up or which are in the market currently doesn't need any skills at all. So it's based on which solution you are looking for. If you are let's say if we are skilled in the, let's say probably the scripting as well as the Linux commands, then probably there are certain tools which is again the FPGA card related tools, or probably developing their own pipeline can be done. If not, then there are cloud solutions which is alternative for people who are very new to informatics or not aware of informatics. So we have both kind of solutions available in here.
Right, sir. Thank you so much, sir. And the other questions we then be taking up via mail, sir. Thank you so much, sir, for your talk and answering the questions. Yes.